Brains and nervous systems of species have evolved to allow individuals to thrive within a very complex world. Now, we can never fully appreciate the outside world, but we can approximate certain features of it through the use of our senses. The skin, as you know, is the largest organ of the body. It's a remarkable organ, really. It's only a few millimeters thick. It gives rise to hair, to whiskers, to claws, to feathers, to nails. It produces vitamin D for us. It has an immune function. It has a barrier function. Of course, it keeps us in. We love the skin. It's something we paint, we wash. It's durable, it's flexible. We tattoo it. We hang ornaments from it. For a neurobiologist, the skin is most remarkable because it harbors our sense of touch. Now, the sense of touch is arguably the sense that we know least about, and that's in part because of the complexity of the peripheral nervous system, and the spinal cord, and the brain to which it projects. The sensory neurons of touch are the longest cells in your body. A mechanosensory neuron that innervates the tip of your toe has its cell body outside the spinal cord and has a central projection that projects all the way up to the brainstem. That's a cell that can be 6 feet long or longer. Now, the skin has enamored neurobiologists for centuries. If you peer beneath the surface of the skin, you see a wide array of sensory endings. The endings, let me give you a little bit of anatomical organization here. The cell bodies align outside the spinal cord in ganglia. There are 32 pair of these ganglia. We call them dorsal root ganglia. I'm going to show you a cross-section. If you look at the ganglia, again, there's about 10,000 neurons in this ganglia. This is the cell bodies. These neurons have pseudounipolar axons that project to the skin or other internal organs, and a central projection that projects into the central nervous system. Now, we're going to focus on these neurons, and the first characterization of the properties of the sensory neurons of touch can be traced back over 80 years to Zoderman and colleagues in the 1920s. That's right about the time when amplifiers were developed through some of the technology that came about after World War I to amplify the miniature electrical signals one can see that propagate from the skin all the way into the central nervous system. This is an electrical signal that propagates at about 200 miles per hour, quite fast. And recording from nerves, one can see that there are a number of different types of touch neurons. We call them low-threshold mechanoreceptors for the aficionados. This is a neuron that responds to stimulating the skin with a force of about 10 millinewton or less. We now know through the work of Ed Pearl and others, there are neurons that are specialized for detecting painful stimuli, and of course, neurons that detect temperature or itch compounds. Sometimes the line between these types has been blurred. Okay. So if you ask any card-carrying somatosensory neurophysiologist, how many sensory neurons in the dorsal root ganglia are there? How many does a mammal have? And they'll tell you that it's probably over 15, maybe over 20 different types of sensory neurons whose cell bodies are within these ganglia and populate the skin with their axon terminals. So I'm fascinated by this question. How many neurons are there? What gives rise to the unique properties of these neurons? What are the unique functions? And how is that information processed in the central nervous system to give rise to the perception of touch and pain? Okay. So our working hypothesis over the last many years, and we're not alone, is that the neurons that comprise this ganglia are distinct not only in their physiological properties, but in their molecular properties. They express different genes, and the expression of these different genes gives rise to their different anatomies and their different response properties. So we and others have exploited this finding. For example, the red neurons are expressing a particular group of genes that allows that neuron to become unique. And so we can sequence the gene expression events in each of these cells by something that's become quite popular these days, single-cell sequencing. And in doing so, one can find that there are cells, when grouped together based on their patterns of gene expression, they cluster together. We define these as cell types. And what's appealing about this for us, this is the work of Nikhil Sharma, a postdoc in the lab, is that it approximates the number of neurons that the neurophysiologist had suggested are present, between 15 and 20 different types. We count about 18 types, depending on how you separate them. Okay, so we also use mouse as a model system, and I should say that the mouse has, as far as we can tell, all of the same types of sensory neurons that we humans have, so we think it's a good model system to study these neurons. Importantly, the awesome power of mouse molecular genetics allows us to exploit what we know about gene expression patterns to express genes like fluorescent proteins and particular subtypes of neurons to understand their properties. So, for example, we can label this one low-threshold mechanoreceptor subtype, happens to be right here, with a particular gene, and study that population. I should say that this population of neurons projects to hairy skin, which is the vast majority of the skin of most mammals. We can label it with a red fluorescent protein, for example, and when one does that, you can then zoom in with your microscope, and Brendan Lennart and Nicole Newbarth in the lab figured out a way where they could actually, under the microscope, put a little electrode in there. Now we can do targeted electrophysiological recordings of these neurons to listen in on their electrical activity to ask what they care about in response to tickling the skin. Okay, so here's an experiment done by Brendan. Part of Brendan's greatness is he's been able to provide quantitative modes of stimuli to the skin. That's been a challenge in the field. He can stroke across the skin. We can puff air onto the skin. And when one does that and records from these neurons, you see that this particular class of neurons responds very well to a gentle air puff. In fact, you can puff air on the skin, on your own skin with this stimulus. You can barely feel it, but these neurons respond. And they also respond to stroking. You can actually take your forceps and deflect a single hair follicle, and this neuron will love that, and it'll fire at 200 miles per hour, multiple action potentials. So these guys are really sensitive to deflecting hairs. And in fact, if Brendan removes the hair from the animal, the hair shaft, they're no longer responsive to this light air puff. They still respond to stroke, which is indenting and moving across the skin. Okay. So why is it that these neurons are so ultra-sensitive to deflecting a single hair follicle? I'm going to tell you a little bit more about the hair of mice. Turns out mice have 9 types of hair. If you ask a mouse hair aficionado, we're concerned with the trunk. There are 3 main types. This big guy here is the guard hair. It's also called the tylotric hair in cats and rabbits. Here's our friend the mouse. These large guard hairs are these hairs here that stick out beyond the surface of the animal. These are really sensitive hairs. The whiskers are something I'm not going to talk about today, enormously sensitive. Almost all mammals have whiskers except for us humans, although if I had any say in evolution, we would have them. These are neurophysiologically remarkable. But let's focus on the guard hair for now. So if you look at the guard hair, here's what it looks like under high-power confocal microscopy. Here's the hair shaft, and the neurons that I'm talking about are these red neurons here. They form what we call lanceolate endings that wrap around the hair follicle. There are 2 other types I'm not going to talk about. We've studied this one in great detail. It forms these circumferential endings. This neuron is really sensitive to pulling on hairs and stroking across the skin. So form is underlying function here. If you look at closer detail, this is the same neurons that I just talked about, the hair shaft group in here. Every one of these large hair follicles receives innervation by 3 different neurons. That's why they're colored in different colors here. And I'm going to zoom in even more because we have a fancy electron microscope, and I'm going to show you what it looks like if we just look at these 2 lanceolate projections right here. So here under EM is the relationship between these lanceolate projections in cross-section and the hair follicle. Here's the basement membrane of the hair follicle. And what I think you can appreciate is these little openings here that come very close, in close proximity to the hair follicle. If you look a little higher here, it's about 100 nanometer distance between the hair follicle and the naked membrane of this, which is the largest cell in your body. And if we look under different conditions, we can actually see little projections protruding from hemidesmosomes in the epithelial cell that come in close contact, and we imagine that these are connecting to mechanically gated ion channels, for example, piezo-2, which Artem Potoposhian has recently discovered, and I think Artem is here today. And our model is that when the hair moves just a little bit, it tugs on these proteinaceous filaments, opening these channels. That leads to the flux of ions through, the excitation, and the action potential initiation, which propagates all the way into the central nervous system. Okay, so form underlies function. This is the population that we're talking about. Virtually every one of these forms this kind of ending. Another type I wanted to just briefly mention is this green neuron. It's actually quite similar in many ways. This neuron, we know, also responds to deflection of hairs. It does not innervate the guard hair, it innervates all the smaller hairs of the body. And here are the endings. So that's all of these small little hairs. And these neurons are incredible in that, every one of these neurons innervates about 30 to 35 hair follicles, and you can see the lanceolate projections. Now what you might notice is these projections are polarized. That's not the case for this other class that I told you about that wraps around the entire hair follicle. These guys wrap only around half the hair follicle. That led us to think that this may be important for a neuron that detects the difference in direction of movement of the hair follicle. So these guys are concentrated on the caudal side and not the rostral side. My postdoc said some people in the audience aren't going to know what caudal and rostral means, so I put tail and head. The head's the part of the animal with the eyes and the whiskers. And so we've recorded, and when I say we, I mean a talented electrophysiologist, Jeff Woodbury. And what Jeff's done is ask, are these neurons in fact responsive to differentially sensitive to hair deflection? And here's the result. Jeff can move these hairs in one way or the other, and you can see they respond much better in the rostral direction movement than the caudal direction. And this is a very talented experimentalist. Jeff can actually go in and with a forceps deflect one hair follicle in each of the 4 cardinal directions. And when he does that, you can see this particular neuron responded best in this direction. Here's another one. It responds best in the rostral direction and only a little bit in the caudal direction. So these neurons are directionally selective. And we wanted to know if this morphology underlies that unique feature of these neurons. And so we asked the question, how does this polarization come about, and can we disrupt it? And it turns out these neurons express a receptor that we've long studied called TrkB. This is a growth factor receptor. The ligand for this receptor is a growth factor called BDNF. And the reason I'm telling you this is because if we look where BDNF is expressed developmentally, it's expressed on the caudal side of every hair follicle. And I think you can appreciate that these nascent developing endings of this neuron grow right where that BDNF protein is. So this is a ligand receptor interaction that's essential for the development of this orientation. And if we make a mouse that lacks BDNF in the skin, these endings end up circling around the entire hair follicle, so you lose this polarization, and then those neurons are completely indifferent to the direction of movement, so you lose the direction selectivity. So the model is that BDNF, because of its polarized expression, leads to the polarization of these endings. These endings are sensitive to hair deflection, more so in one direction than the other, and that developmental event underlies this unique feature of the direction selectivity. Okay, so like the RA neuron I told you before, form underlies function. That's the thought I want to leave you with. today. These two neurons are very similar in that they respond to hair deflection, but by really diving in to understand their properties, which has been made available through molecular genetic approaches, we can understand the differences here. Now, each one of these neurons tells a story. So it's a fascinating, exciting time to be in this field. I should say all of these neurons over here are pain-sensing neurons that are nociceptors. These neurons here are the neurons that respond to itch compounds. One of these populations down here is a neuron that responds selectively to cold. Each one of these neurons is different. It's unique. It's got a unique morphology. In the periphery, it's got a unique pattern of connectivity in the central nervous system. For the touch neurons, which I'm particularly interested in, there are at least 7 types. We've got a molecular genetic handle on 6 of the 7. We're trying really hard to get that 7th one, but they form things like you might have heard of out of textbooks. Here's the Pacinian corpuscle, the Meissner corpuscle. This is the neuron that innervates non-hairy skin in the fingertips and underlies in part the heightened acuity, tactile acuity that your fingertips have. But importantly, we want to now focus, and we are focusing mainly in the central nervous system. If you bring this into focus, a question here is how these neurons with different tuning properties because of these unique structures, how their information is integrated at the level of the CNS. And the canonical view in this field is that there's a labeled line kind of projection all the way up to the brain, and that all computations are the result of clever circuits that the cortex has, the mechanosensory cortex. But the emerging view is that much of this information integration and processing occurs at the dorsal level of the spinal cord, analogous to how the retina processes visual information from rods and cones. We now view the spinal cord as the equivalent of the retina in that sense. Okay, so this is a major focus, both for understanding pain information processing and touch information processing and pain information processing. So ultimately, we hope that this understanding of the sensory neurons of touch and the central circuits they engage will give us a greater understanding of the perception of form and texture, also understand the affective component of touch. And I want to thank our friend, the humble house mouse, for allowing us to use molecular genetics to provide insight into these questions. And I think that in the end, we're going to learn not just about the mouse, but because these neurons are conserved, we're going to learn quite a bit about discriminative touch in larger animals, sensory motor feedback, and the affective component of touch. So I want to thank the mouse for that. I also want to thank our financial support, notably the Howard Hughes Medical Institute, which has had provided long-standing support from my laboratory, and really none of what we do now would have ever been possible without the support of HHMI and NINDS. I want to thank long-term collaborators. I'll point out a couple. Jeff Woodbury, a talented electrophysiologist, my long-term collaborator and friend Alex Kolodkin. One of my scientific heroes, Jack Griffin at Johns Hopkins, really was instrumental in getting us off the ground in some of the directions we're currently taking. Most importantly, I want to thank and acknowledge the members of my lab, both past and present, for their brilliance, dedication, and hard work. Thank you very much.
Questions?
David, that was great. Form follows function. Function follows form, sorry. What is the connection between the caudal expression of BDNF and the curvature of the hair, which also seem to be systematically pointing in the caudal direction? Is that how the BDNF is localized?
So, great question. Hair follicles are polarized. There's a sebaceous gland that's always on the same side of the hair follicle. We find BDNF is always expressed on the same side. If one does manipulations that changes the orientation of the hair follicle, BDNF follows that orientation. So there's something intrinsic, if you will, about the hair follicle we don't understand. It's BDNF transcription that's polarized, I should say, and the fact that a reporter that we use is a readout of transcription, not of protein. So there's something about the intercellular interactions within the follicle that lead to polarization of transcription of the BDNF gene on that side. We don't know what it is.
All right. Thank you, David.
Thank you.